Pamela has developed a specialty mainly in toxicology at the University of Reims and also at the Lebanese University. And she has worked and got a great expertise in the characterization of bioelastic transporters, EPA-G cells, and the investigation of drug-induced cholestatic mechanisms. And today, she's precisely presenting the talk on drug-induced cholestatic liver disease. And we are really would like to welcome her because she's also working with Biopredict International, isn't it, Pamela? Yes?
Yes, it is. Yes, right.
We were also very willing to have this meeting today with you and to hear your presentation, because this is a very special area. Everybody talks about or know more about experimental models in hepatocellular damage, but very little is heard about biomarkers development or even experimental models in cholestatic diseases. So the floor is yours. We really are looking forward to listen to your presentation. Thank you.
Thank you. Thank you for this introduction. So today in this webinar, we are going to see what are the models that can be used to predict drug-induced cholestasis, what are the biomarkers, and what are the industrial challenges in this regard. Let me share my screen. Can you see my screen now?
Yes, very well.
Okay, so my talk will be...
Sorry, Pamela, tu peux connecter your video?
Oui, et quand il y a des vidéos très lourdes que je vais projeter, par contre, je vais arrêter la caméra parce que ça risque de bugger.
Ah,
on est d'accord?
D'accord, ok.
So my talk today will be divided into 4 parts. The first one will be about drug-induced liver injury and drug-induced cholestasis. Then the models that are used to predict drug-induced cholestasis. We will see the biomarkers that the toxicologists are currently using to reveal the cholestatic potential of a drug. And at the end, the industrial challenges that may arise. So drug-induced liver injury accounts for more than 50% of acute liver failure, and they represent the major cause of drug attrition and withdrawal from the market. It is often classified as intrinsic and idiosyncratic toxicity. Intrinsic toxicity is dose-dependent and is reproducible in animals, whereas idiosyncratic toxicity is an unpredictable toxicity that occurs in certain susceptible patients. The main types of lesions...
Sorry, we don't see your screen currently.
Okay.
Neither do I.
Just one moment.
It disappeared after the French lesson.
Tu l'as partagé ton écran?
Là, c'est bon?
Oui, c'est bon.
Now it's coming. Perfect. Yes.
It's good.
On voit les screens et tout vidéo. C'est parfait.
D'accord.
Ça va?
Bon. Alors, yes, I said that the intrinsic toxicity is dose-dependent and can be reproduced in animals, whereas the idiosyncratic toxicity can occur only in certain susceptible patients. The main types of lesions of drug-induced cholestasis are hepatitis, steatosis, and mainly cholestasis. Cholestasis is any situation of impaired bile acid flow because of an alteration of production or excretion of bile acids that result in bile acid accumulation in hepatocytes or in blood. When bile acid accumulation occurs within the hepatocytes, the cholestasis is said to be intrahepatic cholestasis, while extrahepatic cholestasis consists of an accumulation of bile flow from the liver into the intestine. Drugs can induce both intrahepatic
Pamela,
and...
si tu peux, désolé, si tu peux mettre des écrans complets, s'il te plaît.
Oui, oui, je vais essayer.
Voilà.
C'est bon?
Voilà, c'est parfait, merci.
So drugs can induce intrahepatic and extrahepatic cholestasis, acute and chronic, mixed or not with hepatitis. In most of the cases, cholestatic molecules are known to inhibit BSEP, which is a major bile acid transporter. So bile acid synthesis is a complex process that starts from cholesterol and involves many cytochromes such as 7A1, 8B1, and 27A1 to produce primary bile acids, cholic and chenodeoxycholic acids, and secondary bile acids, deoxycholic and lithocholic acid. Both primary and secondary bile acids can be conjugated with either taurine or glycine. So bile acids are synthesized in hepatocyte as well as other bile components. They are excreted in the bile canaliculi and stored in the gallbladder and released in the duodenum. 95% of bile acids can be recaptured by hepatocytes, and on the sinusoidal membrane of the hepatocytes, we have influx transporters, and TCP is the major influx transporter. Efflux of bile acids into the bile canaliculi is mediated by canalicular transporters. The main canalicular transporters are BSEP and MRP2. MRP3 and MRP4 are other transporters, bile acid transporters, but they are localized on the basolateral membrane of hepatocytes. When the canalicular excretion of bile acids is altered, bile acids accumulate in the hepatocytes, and we have the cholestasis context. The bile acid accumulation in hepatocytes can have a direct or indirect effect on the expression of many transporters, like a decrease in the expression of influx transporters, an inhibition of bile acid synthesis, as well as an upregulation of basolateral transport. So in this part, I will give an overview of currently used models to study drug-induced cholestasis. Let's start with in vivo models. So three main models: the genetically modified models, the chemical-induced rodent models, and the combined models. The genetically modified models consist mainly of BSEP knockout mice. This model allows to study the effect of BSEP impairment or impaired activity. The disadvantage of this model is that it represents just one transporter, so it's not really suitable to study the effects of drugs on other transporters. The chemical-induced models are usually created by exposure of animals to chlorpromazine, an antipsychotic drug, or cyclosporine A, a drug used to prevent graft rejection. And it's very easy and rapid to create these models, but the acute effects in these models are really very limited. Plus, we have a large inter-individual differences. Combined models from BSEP knockout and cholic acid feeding are good models because in addition to simulate the BSEP impairment, they develop severe cholestasis. However, the main limitation of all in vivo models is the interspecies differences. In fact, this gap between rodents and humans is mainly due to the more hydrophilic properties of bile acids produced in rodents compared to humans, to the differences in the substrate specificity, and to the alterations in metabolic toxification. So more accurate predictive models are highly needed. The in vitro models consist mainly of primary human hepatocytes, HepA-G cells, HepG2 cells, and iPSC-derived hepatocytes. Let's start with primary human hepatocytes. As you may know, they represent the gold standard, but the main drawbacks of this model is the rapid loss of hepatic functions, the lack of proliferation, and the interdonor variability. Plus, the availability of this model is becoming more and more challenging. Another model is HepA-G cells. HepA-G cell line consists of progenitor cells that are able to fully differentiate into hepatocytes. They express the major metabolizing enzymes and the hepatobiliary transporters. Plus, they can be maintained in culture for up to 4 weeks, so they can be used for repeated exposure and for chronic treatments. By contrast, they need a relatively long period of time to differentiate and to be ready to be used in toxicological studies. HepG2 cells are commonly used because they are easy to handle and highly available. However, they display an extremely low to absent expression of metabolizing enzymes and of hepatobiliary transporters. The iPSC derived hepatocytes can be generated easily from any type of cells and differentiated into hepatocytes without need to a liver biopsy. But the major limitation is that they present a fetal-like phenotype more than a mature phenotype. So their use or their utility in drug-induced cholestasis remains really questionable. All these cell types can be cultured in 3D systems that are supposed to mimic the in vivo situation and to promote cell-cell and cell matrix interaction. So in primary human hepatocytes, the hepatic functions can be preserved for up to 7 weeks, and the cells are more sensitive to hepatotoxicants as the EC50 values observed for many drugs are lower than those observed in their 2D culture counterparts. In addition, it is possible to co-culture human hepatocytes with non-parenchymal cells. The main disadvantages are almost the same for all 3D culture, which is the difficulty to handle the spheroids and the difficulty to make high throughput screening. Regarding 3D HepA-G system, some hepatic functions like albumin secretion, urea production are improved. No major changes of enzyme expression are noticed, most probably because the basal expression is already high in 2D models. And as for primary human hepatocytes, they are more sensitive to hepatotoxicants in 3D system and can be co-cultured with non-parenchymal cells. Concerning the HepG2 cells, 3D cultures slightly improve their hepatic functionality, and data about sensitivity to hepatotoxicants are contradictory, and they can be co-cultured with NPCs. For iPSC-derived hepatocytes, even with the slight improvement of their hepatic properties in 3D cultures, their fetal phenotype remain really a considerable hurdle. In this study, the authors characterized the global gene expression profiles in several human hepatic cell lines, including HepA-G cells and HepG2 cells, in comparison to iPSC and human hepatocytes. Among all the tested models, HepA-G correlates the most with primary human hepatocytes. As you can see here, different donors of primary human hepatocytes constitute a cluster, and HepA-G cells is the closest model to primary human hepatocytes when compared using a list of hepatotoxicity genes. Indeed, the expression of cytochromes in HepA-G cells is in the range of that in primary human hepatocytes, except for CYP2D6, which that is weakly expressed in HepA-G cells. The same can be observed with genes related to liver metabolism transporters and nuclear receptors. So HepA-G is the closest model to primary human hepatocytes. So let's recap this part. The in vivo models do not represent the most suitable model to predict drug-induced cholestasis because of the interspecies differences. The in vitro models, HepG2 and iPSC-derived hepatocytes do not also represent the best models because of their limitation. So primary human hepatocyte spheroid and HepA-G model are really suitable for predicting drug hepatotoxicity. So in the rest of this webinar, I'll talk about HepA-G cells and drug-induced cholestasis in comparison to primary human hepatocytes. And we'll start with the timeline differentiation of HepA-G cells. So HepA-G cell model derives from a hepatocholangiocarcinoma patient with hepatitis C virus infection. It's a bipotent cell line that requires 14 days of proliferation and then 14 other days for differentiation. At the end of the differentiation phase, we can distinguish two different cell populations, the hepatocytes that you are seeing here, the hexagonal shaped cells, surrounded by the biliary cells that are flat and larger. So immunolabeling shows that HepA-G cells express the enzymes, the cytochromes that are involved in bile acid synthesis. And interestingly, these enzymes are only expressed in hepatocytes, but not in biliary cells. Endogenous bile acid content was measured in human hepatocytes and HepA-G cells. And HepA-G cells, and we found that HepA-G cells synthesized bile acids at a rate comparable to primary human hepatocytes. We aim to compare the bile canaliculi structures in HepA-G cells and in human hepatocytes. Photos here show that the bile canaliculi structures in HepA-G cells are similar to those in classical culture human hepatocytes and sandwich culture human hepatocytes. The phalloidine labeling show a pericanalicular distribution of F-actin, and under electron microscopy, we can see that the bile canaliculi in HepA-G cell is surrounded by tight junctions. So then we assess the bile canaliculi surface and HepA-G cells and human hepatocytes, and we found that the mean surface of bile canaliculi in HepA-G cells is similar to that in classical culture with human hepatocyte, but it was larger in sandwich cultured human hepatocytes. We immunolabeled the main hepatobiliary transporters in all three models, and we found that anti-CP was localized to the sinusoidal domain of hepatocyte membranes. MRP3 was localized to the basolateral membrane in all three models, whereas BSEP, MRP2, MDR1, and MDR3 were localized to the canalicular membrane. And again, the expression or the localization of all these transporters was evidenced only in hepatocytes, but not in biliary cells. Excuse me. So, activity of NTCP was measured using radiolabeled taurocholic acid and was found to be, NTCP was found to be active in all three models with a weaker activity in Hep-G cells, which is in agreement with the faint localizations that we can see here. MRP2 activity was also assessed using CDFDA in the presence and absence of an inhibitor of MRP2. As we can see in the absence of the inhibitor, we can see a fluorescent bile canaliculi in both cell models. And in the presence of this inhibitor, there is no fluorescence in the bile canaliculi. We also assessed the activity of MDR1 or MDRs in general using GC1. And in the presence and absence of verapamil. Verapamil is an inhibitor of MDR transporters. So the hepatocytes in the absence of verapamil are green because they cannot accumulate GC1 that is excreted via MDR activity, via MDR transporters, whereas in the presence of verapamil, GC1 is no more excreted, and then it is accumulated within the hepatocytes, and the hepatocytes appeared as red cells, as the biliary cells. The BSEP activity was also assessed using taurocholic acid in the cell lysate and in buffers. We evaluated the canalicular efflux, the basolateral efflux, and the diffusion, and we found that these parameters were almost similar between the three models. So now an overview of biomarkers of drug-induced cholestasis. The bile canaliculi dynamics can be highly affected by cholestatic drugs. So let me please, excuse me. Okay, so time-lapse microscopy, as we are seeing in this video, allowed to reveal spontaneous rhythmic motility of bile canaliculi of Hep-G cells. This motility is characterized by repeated opening and closing spikes every 20 to 30 minutes. These spikes allow dynamic evacuation of canalicular content from saccular bile canaliculi into tubular bile canaliculi. Comparison with the classical cultured human hepatocytes showed similar rhythmic movements as we can see here, while in standard cultured human hepatocytes, also it is possible to see these cycles. Spikes are not detectable. When the spike rhythms were analyzed in the presence of two cholestatic drugs, chlorpromazine and fasudil, we noticed that chlorpromazine induced a permanent constriction of this saccular bile canaliculi, whereas fasudil induced a strong dilation, noting that both chlorpromazine and fasudil led to total disappearance of spikes and a loss of connections between the saccular and the tubular bile canaliculi. Bile canaliculi were further assessed in the presence of different compounds, while some, such as troglytazone, induced bile canaliculi constriction, others like bosentan and diclofenac induced bile dilation. The effect of cyclosporine was dose dependent. It is possible also here we studied the canalicular surface. We assessed the canalicular surface in the presence of cholestatic drugs, and the drug-induced constriction appeared to be irreversible, whereas the drug-induced dilation can be reversible, as we can notice here with tolcaptan. To go further in studying bile canaliculi dynamics, a study has shown the involvement of Rho kinase and myosin light chain kinase signaling pathway using HEPA-G cells in these bile canaliculi dynamics. Briefly, the phosphorylation of myosin light chain 2 allows contraction of bile canaliculi, whereas the dephosphorylation leads to relaxation. So Sharanek et al. Demonstrated that untreated cells showed altered regular phases of phosphorylation, dephosphorylation, phosphorylation, dephosphorylation of myosin light chain 2 every 30 to 45 minutes, indicating a role of myosin 2 in controlling bile canaliculi dynamics. Treatment with chlorpromazine induced, as you can see here, permanent myosin light chain 2 phosphorylation, which explains the permanent constriction observed in chlorpromazine exposed cells. By contrast, fasudil permanently inhibited the phosphorylation of MLC2, which also explains the strong dilation of bile canaliculi in fasudil exposed cells. The effect of a large set of cholestatic and non-cholestatic drugs on the phosphorylation of MLC2 was analyzed. Western blot analysis of MLC2 showed an increased phosphorylation with drugs known to induce bile acid constriction and a dephosphorylation tendency with dilatators. All non-cholestatic drugs did not significantly affect the phosphorylation or dephosphorylation of MLC2. Other biomarkers of drug-induced cholestasis include oxidative stress and mitochondrial dysfunction. Generation of oxidative stress was detected in hepatocytes as early as 15 minutes after chlorpromazine treatment, as shown here by DHE staining, and was totally prevented by the addition of NAC, or the antioxidant molecule. Moreover, the expression of 3 oxidative stress-related genes was induced in chlorpromazine-treated HEPA-G cells and was prevented by the addition of NAC. RAS generation was associated with altered mitochondrial membrane potential, as evidenced by the green color of GC1 within the hepatocytes. This impairment was also prevented by NAC co-exposure. Gene expression and activity of transporters constitute other informative biomarker of drug-induced cholestasis. So here we assess the efflux of taurocholic acid following 30-minute exposure to chlorpromazine. A decrease in canalicular efflux of taurocholic acid was observed and resulted in an intracellular accumulation of this bile acid, whereas basolateral efflux remained unchanged. Taurocholic acid was then assessed at different time points of chlorpromazine exposure. Inhibition of efflux was observed starting 30 minutes with a maximum inhibition at 2 hours. Efflux activity was restored by co-exposure to NAC. In addition, NTCP activity assessed by measurement of taurocholic acid uptake was inhibited starting after 24 hours exposure to chlorpromazine. This inhibition was not prevented by the addition of the antioxidant NAC. We also did a transcriptomic study to assess the effect of chlorpromazine on the mRNA expression of main bile acid transporters and metabolizing enzymes. Chlorpromazine inhibited the expression of NTCP and BSEP in agreement with decrease. Also inhibited MDR3 and CYP8B1, but induced the expression. The downregulation of NTCP that is involved in the influx, and CYP8B1 that is involved in bile acid synthesis, together with the upregulation of MRP4, could be considered as compensatory mechanisms and protective response against chlorpromazine-induced cholestasis. The first biomarker of drug-induced cholestasis is bile acid accumulation. To study the accumulation of toxic bile acids in the liver in drug-induced cholestasis, a study using a large set of cholestatic and non-cholestatic drugs investigated the accumulation of bile acids in hepatocyte cells after exposure to a mixture of exogenous bile acids. As we can see, only the cholestatic drugs cause cellular accumulation of bile acids, while non-cholestatic drugs showed comparable levels of bile acid accumulation to those in control cells. Importantly, this cellular accumulation was characterized with more unconjugated bile acids than conjugated bile acids. Cellular accumulation of the 3 hydrophobic bile acids, deoxycholic, chenodeoxycholic, and lithocholic acids, and their unconjugated and unsulfated forms was observed with all cholestatic drugs, noting that the intrahepatic accumulation of hydrophobic acids is considered as a hallmark of cholestasis. Importantly, while LCA was not detected neither in control or in non-cholestatic drug group, it became accumulated in its unconjugated form after exposure to cholestatic drugs. Hence, the lithocholic acid can serve as a robust biomarker to flag cholestatic drugs. Endoplasmic reticulum stress can be a feature of drug-induced cholestasis. So we demonstrated using cyclosporin A that in addition to oxidative stress, ER stress is a major cause of cholestatic effects of this drug. 4 ER stress-related genes were strongly upregulated after 6-hour exposure to high concentration of cyclosporine. The induction of ER stress was also analyzed after antibiotic exposure using thioflavin T. An early onset of misfolded protein accumulation was revealed in HepaRG cells and primary human hepatocytes, as we can see here, after exposure to cholestatic antibiotics, but not after exposure to non-cholestatic drugs. ER stress sensors of the unfolded protein response were then investigated. Western blot analysis showed 2 to 3-fold enhanced phosphorylation of 2 ER stress markers. In agreement with these results, mRNA expression of ATF4 and CHOP, 2 ER stress-related genes, were upregulated. So far, all the shown results were obtained using 2D monolayer HepaRG cells, but as said before, cultivation in culture in 3D configuration offers many advantages, such as the extensive interaction of between cells and between cell and matrix. So similar to primary human hepatocytes, HepaRG cells can aggregate into compact spheroids that express MRP2 and to a lesser extent BSEP. As in primary human hepatocytes, expression of BSEP was induced upon exposure to exogenous bile acids. Primary human hepatocytes and HepaRG cells were exposed to cholestatic and non-cholestatic drugs in the presence or absence of bile acids for 8 or 14 days. A synergistic toxicity of bile acids and cholestatic drugs was observed at 8 days in both primary human hepatocytes and HepaRG cells and was exacerbated at 14 days. No increase of toxicity was observed with non-cholestatic drugs. Moreover, the known pattern of chlorpromazine-induced cholestasis was recapitulated in primary human hepatocytes and HepaRG spheroids. Indeed, repeated exposure to chlorpromazine resulted in an inhibition of canalicular efflux of a derivative of taurocholic acid and its accumulation into the cytoplasm. Chlorpromazine downregulated the expression of BSEP in both spheroid models and reduced F-actin expression and disrupted its integrity also in both models. An increase in the expression of oxidative stress-related genes was noticed in both spheroid models after co-exposure to chlorpromazine and bile acids. I will conclude this part with the adverse outcome pathway of cholestasis set up by Dr. Winken and his team. So all the known initiating events of cholestasis, such as alteration of bile canalicular hepatocellular change, or change in the activity and expression of transporters can be assessed in Hep-G cells and were found to induce a bile acid accumulation. So two kinds of response are set up, and both can be developed also by Hep-G cells. An adverse one consisting of oxidative stress, ER stress generation, mitochondrial impairment, and cell death, and an adaptive or compensatory response by activating several nuclear factors to recover bile acid homeostasis. The last part of this webinar will be about industrial challenges that are encountered in using mainly Hep-G cell model for predicting drug-induced cholestasis. The first challenge was that Hep-G cells require a long period of time for differentiation. To address this issue, BioPredict has developed cryopreserved, ready-to-use differentiated Hep-G cells, or HPR-116, that can be thawed and used either the same day in suspension or as monolayer, or starting day 7, where the expression of cytochromes is reestablished. As you can see in these graphs, the activities of these 4 cytochromes start to drop after 24 hours of seeding, and then increase again starting day 4 to regain satisfactory values at day 7. The second challenge is to preserve the constant phenotypic features of Hep-G cells throughout cell passaging. As you can notice here, the proliferating cells on one hand, as well as the differentiated cells on the other hand, have the same morphology regardless the passage number. There is also a functional stability between different passages. However, we highly recommend not to use the cells after passage 20, because after this passage, Hep-G cells have shown to lose their conventional morphology and properties. And the main challenge here is to select the appropriate culture medium for proliferation, differentiation, and specific applications. And the most challenging component of the culture medium is obviously the serum. The choice of this latter is of high interest because it can highly impact the morphology as well as the functionality of the cells. Here we can see Hep-G cells cultured and maintained in a medium containing a non-convenient serum. As you can notice, the morphology of the cells is quite different from the normal, with large necrotic areas. Following this first morphological evaluation, we assess many other quantitative endpoints throughout passages, if the serum is convenient or not. However, sometimes industrials have to choose among the available teams to perfectly meet the needs. We at BioPredict are working to set up a defined medium to get rid of the effect of the serum on the cell culture. Another challenge is the weak expression of CYP2D6 in Hep-G cells. And to overcome this limitation, we are working to elaborate a CYP2D6 transfection protocol for Hep-G cells. The first results are encouraging. Indeed, the activity of CYP2D6 increased by almost 20 times after electroporation. However, a lot of work is still required to refine this protocol. One other challenge was to develop a stable bile canaliculi probe, not just to label the bile canaliculi, but to follow a possible alteration of bile canaliculi dynamics. So Starlight, a sister company of BioPredict, has developed a bile canaliculi probe, the SL21FL02, that remains stable for more than 3 hours. Here is a labeling of bile canaliculi in control spheroids, where we can see the bile canaliculi network. Hep-G cells. As you can notice, no significant fluorescence is observed with chlorpromazine within the bile canaliculi. And that's because of the inhibition of BSEP and the bile canaliculi constriction. The imaging of the whole spheroid and particularly of its core was also challenging. With our collaborators, we developed imaging tools and protocols to image human hepatocytes and Hep-G spheroids. In this video, it is the MRP2 immunolabeling in human hepatocyte spheroids. And among our perspectives is to establish a functional cholangiocyte cell line from Hep-G cells. Diana et al. Developed a protocol to differentiation of Hep-G cells into functional cholangiocyte like functional properties of bile acid transport. Immunostaining showed that the Hep-G derived cholangiocytes, but not hepatocytes, expressed cholangiocyte markers and receptors such as osteopontin and secretin receptors. Interestingly, these cholangiocytes do not express hepatic markers such as HNF4 alpha and albumin. In addition, Hep-G derived cholangiocytes are shown to form polarized and functional cysts, as we can see here that CLF, a substrate of MDR, was transported into the central lumen of the cyst. So it would be interesting to develop more this model to assess certain pathologies, including cholestasis and drug effects. So that's all. Thank you for your attention. Thank you for joining this webinar. I'll be happy to take your questions, if any, in the chat box, maybe.
Thank you, Pamela. Very nice presentation and a great overview. The work that you have done and you are already developing, no, in biopredict. I think in a very important area. I've been very fascinated to see the video with the dynamics of the canalicule. No, it has been really very interesting for me. It's the first time I've seen how that appears, no, and been very, very encouraging. And it's true that bile acid accumulation is a hallmark of cholestasis, but interestingly, when there is a bile acid accumulation in hepatocytes, what you have is mainly an hepatocellular damage. Can you tell me how do you think we should measure bile acids in blood in order to really differentiate, not biochemically, but for the bile acids, definition of cholestasis, drug-induced liver injury?
So if I understand well, you want to know how we can measure bile acids in blood, or what is the concentration of bile acids in blood in case of cholestasis?
Yes, because when we defined the type of liver injury, you have an R value that is the ratio between LT and alkaline phosphatase. Interestingly, when you have a severe or a cholestatic damage that induce bile acid accumulation in hepatocytes, what you have in biochemistry, what you see is an expression of an hepatocellular damage. Do you know what I mean? So I think it will be very important to measure bile acids in blood.
Yes, in case of cholestasis, bile acids were measured in blood and found to be in.
Yes, I've seen your data. I mean that we should do the same clinically.
Yes, but clinically we have other indicators like some liver enzymes such as yes, and all other enzymes that can be measured, and we already measured, not we, but in fact the concentration of bile acids was measured in blood in case of cholestasis, and was found to be in like in micromolars, while the concentration of bile acids within the hepatocytes is in the most clinical features used to predict or to know if we have a clinical case of cholestasis is the alteration of liver enzymes, as well as the other symptoms like jaundice, nausea, fatigue.
Yeah. Yes. Yeah. And the other interesting thing for me, at least, was that you were able to reverse that oxidative stress and mitochondrial injury with the administration of N-acetylcysteine. Can
Yes.
you elaborate a little bit further on this?
Yes, in fact, we noticed that chlorpromazine induced a very early a generation of oxidative stress within the hepatocytes, and we were interested to know if this oxidative stress was a critical factor in cholestasis features of chlorpromazine. So we co-exposed the cells with NAC, the antioxidant, and we found that until 6 hours with the co-exposure to NAC, we can reduce this generation of oxidative stress, so we can reduce the progression of cholestasis within the hepatocytes. However, at 24 hours, we are unable to see this reversibility by co-exposing the cells to chlorpromazine and NAC. And interestingly, this effect of NAC was also observed on taurocholic acid efflux, which means that the generation of oxidative stress is really associated to the inhibition of taurocholic acid efflux, while the inhibition of the uptake of bile acids by inhibition of NTCP is considered as a compensatory mechanism. So this one is not affected by NAC because it's not induced by the generation of oxidative stress. It's rather induced as a protective response against the drug-induced cholestasis.
I don't know if there are other questions in the chat. There are people that are congratulating for your talk.
Thank you. Thank you.
Among the attendees, I have seen to Robert, Jordi.
Yes, I
Robert?
have a question. This
Okay,
is
Ramiro.
Ramiro.
Yes, yes.
Ramiro from Valencia.
Yes.
Thank
Because
you very much.
You have recently published a paper.
Some relation with in vitro cholestasis.
Yes.
No, I have a couple of curiosities. So first of all, thank you very much for the presentation. It's very, very, very good. I'm learning a lot. And I have two curiosities. I mean, you have talked about the circular and linear bile canaliculi, and I don't know if this is let's say something that happens in culture cells, or it's something that has a counterpart in liver physiology. I mean, the circular and the linear bile canaliculi that you see in the PARG cells.
In fact, the saccular and tubular the saccular and tubular bile canaliculi were only observed in hepatocyte cells cultured in 2D systems and in primary human hepatocytes classically cultured, but not in sandwich. So, as we believe that the sandwich cultures mimic more closely the in vitro situation and the spheroids also, and we didn't see these spikes in the spheroids, we think that it's most probably a network that is identical to this one, that is exists really in the in vivo situation more than these kind of spikes and saccular and tubular bile canaliculi.
Okay. Okay. Yes, because when I have been working mostly with sandwich culture hepatocytes, so I didn't see these two types of canalicuria, and it was intriguing to me. Okay, another question. I don't understand. I didn't understand. Perhaps maybe when you talk about bile acid accumulation, did you see very interesting large differences between cholestatic and not cholestatic drugs? Where do these bile acids come from? They are de novo synthesis bile acids, or do you put them in the?
Yes. No, here we use exogenous bile acids because the quantity of bile acids that accumulate within the hepatocytes in cholestatic context is not enough to be detected and to see really the accumulation of hydrophobic bile acids. So to exacerbate the effect of cholestatic drugs, we co-expose cells to cholestatic drugs and exogenous bile acids. And then in this case, we are able to measure the bile acid accumulation, the unconjugated and conjugated forms, and specifically each bile acid apport.
Okay, then I understand that the total amount of bile acids in culture plus cells will be the same. I mean, the only thing that will change will be the distribution. They may have more inside than outside.
Yes.
But
yes.
I see differences when you see the non-cholestatic and the cholestatic. The total amount seems to be much higher in the cholestatic.
Yes.
I don't know. I don't know, because the total amount should be constant because it's exogenously added.
Yes, but the bile acids can be also metabolized in cells. So if here, for example, we are seeing the LCA. The LCA, the total LCA amount should not be the same in case of non-cholestatic drugs and cholestatic drugs when we add the cellular accumulation plus the supernatant, because the quantity of, we are not talking about the exogenous lithocholic acid that was added, because the exogenous bile acids that were added can be metabolized by the cells, conjugated and conjugated, or other kind of reactions. So we should not look to see the same concentration or the same total quantity when adding the intracellular and the supernatant quantities.
Okay, I see. If you will represent the total lithocholic acid will be the same, but only the unconjugated is different, and the conjugated will be the opposite, more or less. I see.
Yes, the total, if the bile acid was not processed to give another kind of bile acid, the total should be this.
Yeah, it looks then that the conjugation is like inhibited by the cholestatic drugs because you have many more unconjugated.
Yes, exactly.
Yes.
Than conjugated.
Okay. I see. Thank you. I didn't understand. Well, and I'm sorry, but my last question is more philosophic. At the beginning, did you say that the inter-individual variability is a disadvantage? But I don't know, because in the clinics, there are many idiosyncratic cholestasis, which is due to human variability. Regarding that, I mean, the HEPARG2 cells, HEPARG cells, I mean, you don't have any possibility to have variability because they come from the same donor. I mean, did you think about that? That it may be interesting to develop more cells from different donors?
Yes, sure. It can be considered. So the inter-donor variability of primary human hepatocyte can be considered as a disadvantage because we cannot reproduce the same results many times, but also can be considered as an advantage because it shows the variability and it allows us to study the different phenotypic features. And HEPARG cells, one of their limitations, yes, it is about one donor. Here we are talking about the hepatocytes of one donor, but the drawbacks to develop and to maintain a cell line, a hepatic cell line, a competent hepatic cell line is really challenging, and it's not easy to develop from any donor a cell line that can have a constant constant morphological features. So yes, it can be an advantage and disadvantage at the same time.
Yeah.
I agree.
Yeah. Of course, yes. Working with 10 cell lines at the same time will be crazy. So thank you very much, and congratulations for this very nice talk. Thank you.
Thank you.
Are there any more questions from the audience? Oh, Leonard. Leo, can you talk? Can you?
Maybe he sent. Yes. Can you okay. Yes. Yes. Sure.
Hello.
I will.
Hello.
Yes. Hello.
Yes, I just put something in the chat because I've just been emailed. We've got another meeting 3 minutes ago. But I had several questions for you, Pamela. It was a great talk. Thank you. You know, I've been using the HepRGs quite extensively as well with, you know, in collaboration with Christoph and others. So I was interested in the, you know, if you were, but you answered my questions as I went along, more or less. So the CYP2D6, is it? You've done some transfections with them, and have you developed a stable knock-in?
Not yet, not stable. The results that I showed here in the presentation are really preliminary results that should be refined, and the protocol maybe should really change. The encouraging thing is that we can induce now CYP2D6 for 24 or 48 hours, but the protocol is not yet ready to be used and to change for a longer time. So we are testing now many protocols. We are testing many preparation, but using lipofaction, using lentivirus to set up a protocol that can contribute to develop a cell line that express CYP2D6 at satisfactory levels.
I see. Right. So are these, have you looked at the gene expression level? Is that very low? Is it just not being?
Of CYP2D6?
Yeah.
And hepatocyte cells? Yes, it is low. It is low compared to human hepatocytes. Here I showed a comparison of the expression of CYP2D6. As you may notice here, all the other cytochromes are almost in the same compared to primary human hepatocyte, except CYP2D6, which is really weaker in hepatocyte cells. So that's why we are working on precisely on this cytochrome. In addition, this cytochrome is also involved in drug metabolism. We know that the most important is CYP3A4, that is involved in almost 50% of drug metabolism, but CYP2D6 has also a good part. I think it is involved in 30% of drug metabolism.
It would be nice to have that resource available. I was just going to say, Christian Guggen-Guizot came to Edinburgh a few years ago, 4 or 5 years ago, to talk about the contraction relaxation in cholestatic induced DILI, and DIC, drug-induced cholestasis, and you know that was really interesting. And I think you showed some of the video.
Yes.
This was measured by video. So just because we're going to do something on this, it just reminded me we have a system called ESIS, which is electrical cell substrate adhesion biosensing, or biosensor, which can potentially measure cholestasis, these biophysical changes, non-invasively in real time, like second to second. So it'd be interesting to look at that again, and with a resolution of a deflection of only down to 2 nanometers. So you're going to pick up minute changes in, contraction of the biocanalicula. So it'd be interesting to discuss that again, because I think Christian retired soon after.
Yes, Christian is no more working now. Yes, Christian Guiuzou is no more, I think, active, no more active on this kind of research, but she's writing reviews and she's finishing many other papers. But I think it would be interesting to try this technique. Why not?
She has grandchildren, I believe. So anyway, so I've got to disappear to this meeting. I'm getting beeped. So, well, thank you again, and see everyone soon.
Yes, thank you.
Maybe in Kaunas, if Mr. Putin lets us go in peace.
Yes, hopefully. Yes.
Okay.
Well, thank you so much, Pamela, and all of you for the questions, for this interesting presentation. And we hope we can count on you next March, the 16th of March, in which we are going to have Professor Jan Hensler, who is from Dortmund, Germany, and very well-renowned toxicologist, and he will address, following your talk, Pamela, the importance of bile axis in paracetamol hepatotoxicity, particularly the drug-induced breach of the bile-blood barrier. So we hope we can follow up learning on this very important topic and very challenging at the same time. Thank you so much, Pamela, for sharing your knowledge and expertise with us this afternoon. All the best.
Thank you.
Keep you in touch.
Yes, thank you.
Thank you, Pamela.
Thank you. Bye.
Bye.